Terima kasih sekali atas kehadiran Titi dan teman dari BPT dan juga yang lainnya, Mahmudun dari S2 TIP dan juga teman lain. Kita akan menikmati waktu mungkin sudah sekitar, I will give you time so until 1 hour if you need
okay.
it.
Maybe
And then extra time and then finally. Yeah. Okay, Pak Ben. Okay. Give applause for him.
No, I don't
oh, okay, yeah.
Madurun sangat. So, ibu-ibu dan bapak-bapak yang saya hormati, maaf saya tidak bisa berbicara bahasa Indonesia, karena itu saya berbicara bahasa Jerman, ya. No, better bahasa Inggris. So, first to introduce myself, why I'm here in Indonesia. So the story happened in the year, now we have to go back to 1987. I just had my PhD for half a year, then my boss came and said, join me to the office, okay, what's going on? And this is good that here was still some fresh tempeh standing, because this makes a good bridge to tell you the whole, Jimmy! Apa kabar? So, then my boss came and said, join me to the office, sit down. Then he said, do you know what is tempeh? And I just looked at him and he said, I can see from your eyes you don't know what is tempeh. But do you know what is biotechnology? I said, of course, because I have to replace you so many times when you're out of the institute. And then the decisive question came, would you also be willing to replace me on a delegation traveling of the German Ministry of Research and Technology? And without hesitating, within a second, I said, yes, where do I have to go to? And he said, to Indonesia. When will I be there? In 2 weeks you will be there. Okay. And then the whole story was concerning Teknologi, and I was going first to BPPT. And the first person I met in there was Pak Suyanto Parviroha Sorno. And I met him yesterday also again. So, and then we are going to several places, also in here to UGM. And in those days, Pak Slamet Sudamaji was giving a lecture, yes. So when I was coming back, then my boss said, Mr. Bisping, do you think it's possible to write a research proposal concerning tempeh kedele? I said, yes, of course. Sit down and write. Yeah, so then I had to sit down and write, and we had a research proposal, but then it had to be corrected a little bit. And we had to go to the ministry, but then the whole program together with Indonesia started. And this was cooperation together with bioindustry of BPPT, and this was starting in April 1988 and was lasting until end of September 1997. And this gave me the chance to do the licensee, what we call in Bahasa Jerman, Habilitation, and this is a driver's license to become a professor. And then later, so nowadays I'm a professor of food microbiology. And why food microbiology? Because tempeh changed my whole life. Because before that I was a person who doing work in technical microbiology, submerged fermentation, but tempeh brought me to solid state fermentation and food. And this is my position nowadays, because nowadays my position is food microbiology and hygiene originally, but I changed the name into food microbiology and biotechnology. And then from 2002, for 6 years I had cooperation again with BPPT, but then in the working up of the kulit udang as a basis for chitin as a precursor for chitosan. And then later we had a project together with Bacterium, and this was on enrichment. Yes, I'm Dick Benz, Bacterium. So we had the project together on tahu, enrichment in bacteriocin, and on the other hand side, enrichment in vitamin B12, because in the young generation in Europe, many young people decided to become vegetarian or even vegan, and those people, if they do not supplement vitamin B12, maybe after 5 or 6 years, they have problems in their health from anemia that will be developing. Okay. But now we have a different topic, and the new topic is cacao fermentation. and I have a cooperation in the moment together with Peru, and I just returned from Peru because 10 days ago I still have been in Peru on the Salon de Cacao y Chocolate and have been to some cacao farms in there. So, but now the question was the topic of the research with Peru. Do specific yeast strains have a capacity to influence cocoa aroma during fermentation? So, what's the introduction? Let's say look for the cultivation of cacao and their varieties. So Theobroma cacao grows in the tropical area. So here you see the tropical belt in between northern and southern circle of latitude, with varieties originating from the forest areas of South America. And here we are in Peru and Ecuador. And that's the original origin of cacao. So we have 4 different varieties, but these varieties are only in the trading. So the traders differentiate between Criollo, meaning this is fine flavor cacao, and Criollo meaning the original, the native one. Then we have Forastero, meaning the strange one, and this is Bal cacao. Then we have Trinitario, and Trinidad is in here in the Caribbean. And this is flavor cacao, and this is a hybrid between Forastero and Criollo, because there was what also in here happening from time to time, eruption of a big volcano destroying all the cacao plantation, and from the rest that were there, then they did the hybrid of the Criollos and the Forasteros. And in Ecuador we have a trading variety called Nacional, so meaning this is flavor cacao from Ecuador, and Ecuador is located here in the north of Peru. Okay, but if you look into the genetics, we can nowadays differentiate about 12 different different types that are there. Okay. So, in here you see that the cacao fruits are growing directly on the stem, and this is a specialty. Okay, and if we look in here, we have from the colors and from the forms a big variety of different genetical types of cacao, and what the aim of the whole story is a high quality of the products of cocoa and chocolate. So, now concerning fermentation. In the beginning we have the cacao pod and we do the fermentation, but the fermentation is always a spontaneous fermentation, meaning we have the introduction of microorganisms by opening the pot, and we have introduction by the big knife, the so-called maquete, and by the hands of the co-workers, and of course everywhere where we have something sweet and a little bit acidic, insects are attracted, and the insects are the vectors for introducing the microorganisms. So then after fermentation the cacao seeds are dried, and then we have the cacao bean. And you have to know, in literature the terms cacao and cocoa are always mixed up. And I decided for myself just to be strict, following the dictionary Merriam-Webster's of the US, and it says everything before the roasting process is cacao. Only after the roasting it's called cocoa. But in literature it's always mixing up. So after roasting we have cocoa, and then we can do the production of the chocolate. So, but the quality of the raw cacao, this is wrong term here, is highly variable, depends on the fermentation. And the spontaneous fermentation is the first stage in the production of cacao and chocolate, and we have to have a special succession of microorganisms. And the key microorganisms first are yeast, then we have lactic acid bacteria, abbreviated with LAB, and we have acetic acid bacteria. So now in here you see the opened pod, and you see surrounding the seeds we have a whitish pulpa, and this pulpa is containing sugars but is also containing citric acid. So in here in Indonesia we have a big disadvantage because we have traders from Malaysia, and the traders from Malaysia are giving to Indonesian farmers the same price for the cacao, either it's fermented or not fermented. And why should Indonesian farmers do more work if they do not get any money for that? That's crazy. And I was saying this in public 2 years ago in a congress in Germany, and there were also some traders from Malaysia and fighting against me. But I said, that's the true information that I got. Okay, so because of this, you see here in Padang, these are just, too quick. dried on the floor, and here even on old sacks of concrete. But what is much better is fermentation. And here we have a picture from Ghana, traditional fermentation on banana leaf. So the cacao seeds are shoveled up to a heap, and then the leaves are closed, and then they put some stems on it, and then we have an anaerobic step. Okay. So then in here, in Padang area, we have small boxes, and they also put some banana leaves in there, do the fermentation in these boxes. But here, this is from textbook or internet, box fermentation in Malaysia, but much better looking like this. This is from Jember, and this is a picture I took some years ago when I was in Jember. And you see, we have an upper box and a lower box. And in the first upper box, we have the anaerobic step, and this is lasting for 2 days. And after 2 days, it's shoveled down to the next box. And by shoveling down, we have an aeration, and the reason behind that you will see soon. Come in, please. Okay. All of you, come in. So, and in here now you see several boxes behind each other, because different varieties of cacao need different length of fermentation. And in the area in Peru where I've been 2 years ago, so this is in the middle of the jungle, there they have the really original cacao, and this is called chuncho. And the chuncho needs only a very short fermentation time of 4 days. 4 days. But if you have the other ones like CCN51, they may be taking in between 5 to 7 days, depending on the situation. So then after the fermentation, we have to have a drying process, and this mostly is done under the sunshine, or in here we have also an artificial drying. And then first we have sun drying, maybe for 5 to 7 hours, and then depending on the heat, it takes 6 hours, 8 hours, 10 hours, whatsoever. So, but what's happening during the fermentation? In the first anaerobic step, we have the growth of yeast. So yeast, normally in here it's just called ragi. This I know from ragi tempeh, but then it was Rhizophus strains. So the yeasts are producing alcohol, ethanol. So meaning the first step is alcoholic fermentation, like we have in the production of wine and beer, or like we have in the production of roti or keju. It's also alcoholic fermentation. But the roti or keju does not contain the alcohol anymore because it's baked, and by the heat all the ethanol is evaporating into the atmosphere, and we only use the CO2 that is leavening the dough. Okay, alcoholic fermentation. Then a little bit later, but nearly at the same time, we have the growth of lactic acid bacteria. And they are producing lactic acid. And then the yeasts are degrading the pulpa. And by this, the pulpa is dropping off, and then we have bigger holes in between the seeds. And this is increasing and helping the aeration. So after 2 days, the seeds are shoveled around. So then we have oxygen coming to the whole situation, and this is leading to the place that we have the growth of acetic acid bacteria. So acetic acid bacteria are oxidizing the ethanol to acetic acid. And the English term for acetic acid is vinegar, but the term vinegar is coming from French language, meaning vinaigre. Vinaigre, vin means wine, aigre meaning sour, sour wine. So vinegar is nothing else than sour wine, so oxidized ethanol. And then we have the acetic acid peak, and what's happening then? We have the cacao beans in the big heap or in the box, and like in the decomposing of waste material from plants, it's heating up, and we reach a temperature of up to 50 degrees centigrade, and we have the acetic acid. And under these conditions, the acetic acid is invading the seed via the micropula, and the heat together with acetic acid is leading to the situation that the embryo in the seed is dying off. But at the same time, under these conditions, we have the activation of plant-owned proteases. And these are degrading storage proteins to free amino acids and free short-chain peptides. And these will be forming the precursors during the roasting process that gives finally the aroma to the cacao and to the chocolate. But you see, in the final step there may be also some spore formers, but all in all, the fermentation takes 5 days, in some situations up to 7 days. So what's happening? In here we have the first step in A, pulp degradation. The sugar is by alcoholic fermentation metabolized by the yeast to ethanol, but at the same time the yeasts are degrading the pulpa that is dropping off. Okay. So then we have the step of acidification. So ethanol is oxidized, but we also have lactic acid production, and then the acetic acid invading the seeds through the mucopula. So this is the spot on the seed where normally the radicula, so the first root coming out. And then finally we have the browning process. But you see in here we still have the testa surrounding the seed, and this is still a kind of protection, and you will see later what's the reason behind that. So, what is known from research on wine? So now we go to Teknologi. Why? Because the first step is alcoholic fermentation, and this is very similar to alcoholic production. And now funny story in behind. So when I was first time in Universitas Andalas in Padang, so then I recognized that there are 2 lecturers, Papiri and his wife, and both did their PhD in a university in Geisenheim, with which I'm cooperating now, and this is specialized on winery, the research. And then I asked to the Dean, Bahadinis, how come these are strict Muslim and doing PhD on winery research? What's behind that? I don't understand. And then Bahadinis told me in Al-Quran it's not written you are not allowed to drink anything, but it's written in there you are not allowed to be drunken. That's a difference. And finally Papiri, when I met him first time, was I could not work on winery research anymore. But what was he doing in Universitas Andalas, his new field of research, Tape. We have a similar process. So, but what's known? So the aroma of the grapes, of the fruits, is depending on the variety and the genotype in cooperation with environmental factors. And these are called by the winery people terroir. So, and this means we have an influence of the climate, influence of the composition of the soil, and influence of the topography, meaning the height, the level, and the mountains, whatsoever the area where the plants are growing. So, but the same factors are influencing and are also effective in cacao fruits. So then we have microorganisms, especially yeast, that are influencing the aroma by different mechanisms. And first, the yeast are consuming compounds of the juice and producing compounds that may be influencing the aroma. So then we have compounds with a neutral aroma that are transformed by enzymes of the yeast into aromatic compounds. Because many aromatic compounds, before they get aromatic, they are neutral. Why? Because they are bound to sugars. We have glycosidic bondages, and then these compounds do not have any aroma. Only if the glycosidic bondages are split off, then the aroma is developing. So then we have compounds of primary metabolism of the yeast, like ethanol, but also glycerol. But what is glycerol? Glycerol is a C3 compound, so small molecule with 3 OH groups. And the original meaning of the word glyceros in Greek language meaning sweet. Because glycerol, if you taste it, put a little bit on your tongue, it's tasting sweet. So meaning, and glycerol is always a normal byproduct of alcoholic fermentation. Meaning, if you are coming back to the example of wine, and people say the wine is completely dry, meaning there's no sugar at all anymore in the wine. But it may still have a little bit sweet taste because of the glycerol. There's always a normal byproduct of the fermentation. So then we have acetic acid. We have acetaldehyde as a precursor of ethanol. And we have metabolites of secondary metabolism like esters, higher alcohols and fatty acids that are produced by the yeast. So then we also have terpenes, esters, aldehydes that lead to aroma and flavors that are described as flowery, fruity, or peppery. And some weeks ago I was listening to a lecture in a guest lecture in our institution, and this was concerning compounds of urine, of pipis, and this was quite strange because the lady talking about that topic said that in the urine, it was concerning some kind of illness, are 5 different small molecules that all have a very bad taste. But if you put these 5 small molecules together in the right ratio, the aroma is like nanas. So, meaning, aroma is not always produced by only one substance, but if you have the combination of different substances, that by itself all are very ugly, but then you combine them, then it gives a good aroma. So, funny thing. Okay. So then we have monoterpenes, and here we have substances like citronellol, so coming from geruk. Then we have linalool, geraniol, can be set free enzymatically out of glycosidic bounds precursors by yeast or some yeast strains, what I told you before. And one example is a yeast called Saccharomyces lactis. But why such a complicated name, Saccharomyces? So in the lab there's also a glass vessel called Kloiverkolben, because this was a scientist from Blanda and he had the family name of Kloiver, so that's the reason of this strange name. And lactis, meaning it was isolated out of the milk, can produce those substances newly. So different yeast strains produce different aroma active substances, and here we have now concentrations given in milligrams per liter. And you see in red we have the yeast Saccharomyces Cerevisiae, but who of the students knows the meaning of the word Saccharomyces Cerevisiae? We always use it, but you should also know the meaning behind it, then it's easier to keep it in the mind. So, what's that? Saccharomyces cerevisiae is the normal beer or wine yeast or baking yeast. But the meaning of the word is Saccharine, meaning sugar, gula. Mykos is the fungus. So Saccharomyces means sugar fungus. Cerevisiae meaning of the beer. So the sugar fungus of the beer. This is Saccharomyces cerevisiae. And here we have another yeast strain called Hansenia spora uvarum. Well, why Hansen? So this was a researcher, microbiologist again, and this spore forming. Uva is the grape. So meaning this yeast was isolated from grapes from the man called Hansen. So but now if we look at the aromatic compounds produced by these 2 different yeasts, you see in here Saccharomyces cerevisiae has a little peak, and here milligrams per liter of up to 200 milligrams per liter of isoamyl alcohol, and we have quite a big peak in 2,3-butyl-diol. But in comparison to that, Hansenia spora uvarum does not have these peaks, but a peak in acetoin. And acetoin is not such a good smelling aromatic compound. Then in here we have another yeast called Saccharomyces ludwigii. So there's the male name Ludwig in the species name. And if we look here, it has again a very different composition of aromatic compounds because it also has acetoin. But then in here a bigger peak in isobutanol. And we have a very big peak in here in ethyl acetate. But ethyl acetate by its own, it's also not a good smelling substance, and all the ladies will know, because if you are putting colors to your fingernails and later you want to get off the colors, you have to take, for example, this substance to get the fingernails pure again and clear again. And I, as a biologist, had to use ethyl acetate in my basic studies, because we had to do for zoology a collection of different bugs, and we were killing the bugs with ethyl acetate. So, what is known from cacao fermentation? So during the fermentation, the yeast produce ethanol and pectinase that are leading to liquefaction and the dropping off of the pulp. So the pectinase is degrading the pectin, then the pulp is dropping off, and we get holes in between the seeds. So then we have Homo fermentative lactic acid bacteria, that are producing lactate. So who of the students is know what is a Homo fermentative lactic acid bacterium? What does it mean, Homo fermentative? Homo means always that something is the same. Homo fermentative meaning the products of fermentation are the same. So, and the products are only lactic acid. So Homo fermentative lactic acid bacteria means these bacteria out of 1 mole of glucose are only producing 2 moles of lactic acid, nothing else. But then we have heterofermentative lactic acid bacteria. Hetero meaning different. Heterofermentative meaning the products of fermentation are different products. And in this case, it can be if we give 1 mole of glucose to the bacteria, they are producing 1 mole of lactic acid and 1 mole of CO2. But we still have 2 carbon atoms that are surplus. And these may be ethanol, and then the energy win is only very low, or maybe acetate. Then the energy win is higher. But the cell has a problem, because in the metabolism it has to oxidize NADH2, has to be reoxidized to NAD+, meaning we need an electron acceptor to produce acids. Again, if we have fructose in the medium and this is reduced by electrons, what's the result? So fructose has a keto function. And what's happening if we are reducing the keto function? It becomes an OH group. And then out of the sugar, fructose, we get a sugar alcohol. And in this case, this is mannitol. Okay. So then acetic acid bacteria are oxidizing ethanol produced by the yeast to acetic acid. Another question to the students to keep you awake. What, besides acetic acid, is the most important substance produced by acetic acid bacteria? And this is very important for all human beings for the nutrition, because it's a vitamin. It's vitamin C. It's always produced by acetic acid bacteria, because in the production of vitamin C we have a step where we have an oxidation, and this is the step of oxidizing sorbitol to sorbose. And this is the precursor for vitamin C, for ascorbic acid. Just to keep in mind what is the, let's say, the main task of acetic acid bacteria. And another thing all of you know, because it's also a well-known product in Indonesia of acetic acid bacteria. But this is only of Acetobacter xylino. What is this one? Nata de coco, yes. Okay. So, then ethanol and acetate penetrate through the microtubule, I told you before, into the seed, and in combination with the elevated temperature leads to a dying off of the embryo. And now plant-on proteases are activated, degrading storage proteins and so leading to the production of short-chain peptides and free amino acids that are the precursors during the roasting process for the cacao aroma. So now we have an example of fermentation done by my former co-worker Fadu Rosi, who was working in Lipi Cibinong. And here we have a fermentation time of 96 hours, and now we look into development of temperature and pH value. And you see in here the temperature is decreasing from 28 degrees centigrade up to 50 degrees centigrade. So then in here the pH value in the beans is dropping off from 6.0 going down to 4.7 about, and in the pulpa it's a little bit varying from 4.0 to 4.3. So then looking into the population of the microorganisms, so and again here in the first 24 hours we have a sharp increase of the yeast, going up from maybe 1 to 2 times 10 to the power 5 up to nearly 8 times 10 to the power 7, and then after the first day slowly decreasing to 3 times 10 to the power 7 and then going down. So then we have lactic acid bacteria, and then going up into the first 2 days, up to 5 times 10 to the power 9, and then decreasing. And we also in here have acetic acid bacteria, and these are going up here to 5 times 10 to the power 8, and then keeping stable. So then we look for the production of ethanol and glycerol in the pulp, and you see in here the ethanol production starts a little bit later, but we here have the growth of the yeast producing ethanol, and we have the peak nearly after 2 days, in here going up to 19 milligrams per gram, or let's say 19 grams per gram of pulp. And in parallel to this we have the production of glycerol, but only going up here to 4 milligrams per gram. So, but within the beans we have an accumulation of the ethanol up to the second day, up to 13 milligrams per gram, and also here the glycerol is increasing up to 1.5 to 2 milligrams per gram. So then changes in organic acids in pulp and beans, and first we look into the pulp, and I told you before, the pulp is containing citric acid. So if we look at this, in the beginning we have a citric acid concentration here of about 17 milligrams per gram, but by the growth of the yeast it's degraded after 1 day, going down to 0. So then we have succinic acid. In the beginning it's very low, but after 2 days we start the aeration by shoveling around, and because of this, succinic acid is always a product of the citric acid cycle, and because of this after 2 days it's increasing. Then we have lactic acid, going up slowly, and we have acetic acid only after the aeration. So no, here is acetic acid going up here to 12 milligrams per gram. So within the beans we also have the degradation of the citric acid, and in here with the dark spot we have increase of lactic acid up to 3.5 milligrams per gram, and the open circle, now important, acetic acid invading the bean, going up to nearly 7 milligrams per gram. So now the question is, if ethanol, glycerol and organic acids are able to penetrate into the seed, why also small molecules like esters and monoterpenes, terpenoids, should not be able to do so in the same manner? So then we have a patent of people from Christian Hansen company in Denmark. So these are called Zarens and Schwiegers, and they said we can enhance the quality of cocoa and flavor by using a yeast strain called Pichia cloyveri. Remember before we had Saccharomyces, also the same scientist in the background, cloyver as yeast starter culture for cacao fermentation. And now you see here in this graph the production of isoamyl acetate, and Saccharomyces cerevisiae is producing here in ppm per gram up to 0.15. But Pichia cloyveri is producing much more, up to 0.23. And another one in here, Candida krusei, producing up to 0.16 maybe. So meaning Pichia cloyveri is producing much more isoamyl acetate than Saccharomyces cerevisiae. So then looking into isobutyl acetate, in here Saccharomyces cerevisiae is producing 0.08 ppm per gram, but Pichia cloyveri producing up to 0.14. And only Hansenia spora diamondii also producing up to 0.13. So meaning we have special small molecule compounds that have an impact on the aroma produced by special yeast strains. And then they did an experiment, and SP in here meaning spontaneous fermentation, PK meaning fermentation with the addition of Pichia cloyveri. And if you look in here, cocoa aroma was produced by both strains. But using Pichia cloyveri we have a higher taste in the acidic direction, but the spontaneous fermentation, the cocoa was tasting more bitter and more astringent than in the fermentation using Pichia cloyveri. But then on the other hand side, using Pichia cloyveri, we have a higher impact of floral aroma and of fruity aroma. Nutty aroma is nearly similar. So meaning the use of Pichia cloyveri in this case leads to a reduction of bitterness and astringency, but leading to an improvement of floral and fruity aroma. So then they had a tasting panel, and as an average score of 2 rounds of tasting by the panel, all the people said, okay, we prefer the chocolate produced or the cacao produced by Pichia cloyveri. And the overall preference was that all testing panel persons preferred the cacao or chocolate produced by Pichia cloyveri in comparison to the spontaneous fermentation. So, what's happening in spontaneous fermentations? And here we have results of Faro Rosi again. And you see here, and this fermentation I think he did in Jamber. In the beginning, the first day, we have an amount of cells, colony-forming units of Saccharomyces Cerevisiae up to 4 times 10 to the power 4. Then we have Hansenia spora Guillemondii up to maybe 2 times 10 to the power 4. Then in here the gray one is Hansenia spora opuntiae up to 1.5 times 10 to the power 4, and same is valid for Candida tropicalis and Hansenia spora thailandica. So this is the starting value. After 1 day, you see in here a big amount of different yeast strains is popping up, but then after 48 hours the whole story is collapsing. So only Saccharomyces cerevisiae is surviving. Then we have Hansenia spora Guillemondii and Torula spora delbrueckii. And then, so this after 3 days, only Saccharomyces cerevisiae left, after 4 days also. And here, after 4 days, also Sporobolomyces roseus popping up. So what this, meaning the yeast population is growing up, increasing after 24 hours, but then suddenly collapsing. But then the question is popping up, what's the reason behind that? There must be a reason that the yeast states suddenly collapse. So, and what's the answer of the literature up to now? This effect is caused by temperature, ethanol and acetic acid. Of course, acetic acid is always a good preservative, killing microorganisms. Ethanol, also different yeast strains have to withstand the ethanol. And also we have the sensitivity against the temperature. So what's said in literature makes sense, but it cannot be the whole answer of the story. So, and then one co-worker of mine, Claudia Barman, and this is one of the persons who now was for 9 weeks in Peru to take samples of cacao fermentations, together with my other PhD student, who is from food chemistry. And she did her PhD in 2014 in the group of Reinhard Lieberei. And Reinhard Lieberei was the professor of tropical crops in our university. And he was the leading, let's say, the Pope or the best professor in Germany on cacao research. And she did her PhD with him, and she found out that the testa of the cacao seeds, after degradation of the pulp, is releasing a slime containing antifungal proteins. And in here you see a cacao seed after the pulp was degraded by enzymes, and then she put the seed with the testa only in sterile water, and then the testa is producing such big amount of slime. So, and then the question was, because for the traders this slime is only waste material that has to be transported. So the companies wanted to know, how can we get rid of the slime? But we want to know, what's the function of the slime? It must have a function, otherwise it will not be produced by the seed. So, and then Claudia looked into this whole story, and you see in here the rest of the testa. Here we have the cotyledon, here we have the pulp, and in here you see some slime cavern. So, and the slime is located directly below the outer epidermis, and it had not been described in detail in literature, and the chemical composition was unknown. And then Claudia reported that this slime is produced for 3 days after removal of the pulp, and the slime is composed out of sugars and different kinds of proteins. And the funny thing is, what she was identifying in the slime were enzymes, and these enzymes was a chitinase, a glucanase and osmotin. So, and this was investigation of activity of the testa associated slime. And you see in here she was working up the slime, doing SDS page, and here we have the ladder. And you see in here in 6 we have a band, and this was identified as osmotine. And osmotine is a protein with an activity against fungi, because it's setting into the membrane and leading to channels in the membrane, and then the fungal cell is died off, is killed. So then we have 7 chitinase of Theobroma cacao. Most of the fungi are having in their cell walls chitin, and if we have a chitinase in the seed, so this is just defense mechanism against fungal attack. And then we also have in here, in number 9, here is a band number 9, L-glucan endo 1,3 beta glucosidase, so also degrading the glucans in the cell wall of the fungi. Meaning the cacao seed has a natural defense mechanism against fungal attack. So, and then Ozzy did some tests, and here in agar diffusion method he was testing the activity of the slime against a yeast called Rhodotorula rubra, and you can see in here they are inhibition zones depending on the different concentration of the slime. And then he also did a micro dilution method, and in here this means we have the yeast only in distilled water, and the optical density is increasing here to 0.45, after 2 days to 0.65, but with different concentrations of the slime it's lowered down. And as a positive control he was taking 2 milligrams per milliliter of propionic acid to inhibit the growth of the fungi, in this case of the yeast. And you see there is an activity of the slime against the yeast cell growth. So then he was testing against Saccharomyces Cerevisiae, and you see the effect is much lower. So now remember the situation after the first day of fermentation. We had a big variety of different yeast strains, and then after 2 days only Saccharomyces cerevisiae surviving. Meaning Saccharomyces cerevisiae is not as sensitive against the enzymes of the slime than other yeast cells. So the big diversity of yeast cells is diminished, so to say, by the enzymes and by the proteins of the testa-associated slime. So then in here there was a testa inhibition test against different fungi and yeast, and you see we said we have chitinase and we have glucanase. But here we have mucor and we have rhizopus and we have Rhizopus oryzae. So all of these are zygomycetes, and Rhizopus oligosporus oryzae you know from tempeh. But these zygomycetes have a different composition of the cell wall than the other molds in here, because they are not containing chitin in the cell wall but chitosan. And then this enzyme has no activity against it. Because of this there is no inhibition. And you see in here also in the yeast cells there are some yeasts that are resistant to the activity of the proteins in the testa-associated slime. So what's the conclusion? Aroma influencing yeast strains have to be identified and characterized, and this is what my co-workers are just doing or were just doing in Peru. So then they must have the ability to grow on cacao pulp and the capability to degrade the pulp, that's necessary. Opala, go back. And then if they are used as a starter culture, they must prevail against autochthonous yeast. So if we are adding a selected yeast that we want to use as a starter culture, we always have the ecological fighting in between the spontaneous popping up yeast cells and bacteria and the yeast cells we want to grow. So this is still a question of research. Is it strong enough? So then they have to be resistant against temperature, ethanol concentration and acetic acid concentration. And they have to be resistant against testa-associated slime, or must have produced aroma-influencing compounds before they are suppressed by the enzymes of the slime. So, meaning there's still a lot to be done in the research of cacao fermentation, And now I want to acknowledge, on the one hand side, Professor Reinhard Lieberei, who was retired since a few years, but he was the leading scientist in cacao research. Then Claudia Barmann, still my co-worker now, who did the first investigation of the cacao slime. Then we have Professor Nicolas Niemenak. And Niemenak is a friend of mine from Cameroon, and he is in Yaoundé University also doing research on cacao. And Dr. Fado Rose is my friend Ozzy from Lipi Cibinong. And thank you very much for your attention. Ori bahasa Indonesia. Terima kasih banyak atas perhatian Anda. And here we are in Jogja. Matur nuwun sanget.